Hello everyone, I am Mohammed Hamama, and this is your ASCP preparation camp. In this camp, we will go through each topic on the ASCP lecture list. Today, we'll be discussing other extrinsic defects leading to increased erythrocyte as hemolytic anemia that is caused by infectious agents, and red blood cell injury. And we will start with hemolytic anemia that is caused by infectious agents. Many people may not be aware that certain infectious agents can actually cause hemolytic anemia, a condition where red blood cells are destroyed at a faster rate than they can be produced. One of the most common infectious agents that causes hemolytic anemia is malaria, a serious and sometimes fatal illness caused by the Plasmodium parasite. The parasite infects red blood cells and causes them to rupture, leading to a rapid destruction of these cells and ultimately resulting in anemia. While Plasmodium falciparum and Plasmodium vivax are the most common types that infect humans, other species such as Plasmodium ovale, Plasmodium malariae, and Plasmodium knowlesi can also cause malaria. Plasmodium knowlesi is a particularly interesting species, as it naturally infects macaque monkeys and can be difficult to distinguish from Plasmodium malariae under a microscope due to their similar appearance. In fact, for many years, hundreds of cases of Plasmodium malariae in Malaysia were actually caused by Plasmodium knowlesi when more advanced laboratory tests like PCR were used. Plasmodium knowlesi malaria is now known to be widespread in Malaysia and cases have been reported in other parts of Southeast Asia as well. Life cycle of Plasmodium. Plasmodium is a protozoan parasite that belongs to the phylum Apicomplexa. It has a complex life cycle that involves two hosts, a human and a mosquito. The human host is where the parasite undergoes asexual reproduction, while the mosquito host is where the parasite undergoes sexual reproduction. The life cycle begins when an infected female Anopheles mosquito bites a human and injects saliva containing sporozoites into the bloodstream. Sporozoites are the infective stage of Plasmodium that can travel to the liver and invade hepatocytes. Inside the hepatocytes, the sporozoites multiply by a process called exoerythrocytic schizogony, producing thousands of merozoites daughter cells. After several days the hepatocytes then rupture and release the merozoites into the bloodstream, where they can infect red blood cells. This stage of the life cycle is called the pre-erythrocytic phase because it occurs before the infection of RBCs. Once inside the RBCs, the merozoites start another round of asexual reproduction called erythrocytic schizogony. They go through different developmental stages, such as ring form, trophozoite, immature schizont, and mature schizont. The mature schizont contains many merozoites that break out of the RBC and infect other RBCs. This cycle repeats itself every 48 to 72 hours, depending on the species of Plasmodium. The rupture of RBCs causes hemolysis and anemia, as well as fever and chills that coincide with the release of merozoites. This stage of the life cycle is called the erythrocytic phase because it occurs within the RBCs. Some species of Plasmodium, such as Plasmodium vivax and Plasmodium ovale, have a dormant stage called hypnozoite that can stay in the liver for months or years without causing any symptoms. These hypnozoites can be reactivated by various triggers and cause a relapse of malaria. Not all merozoites become schizonts in the RBCs. Some of them differentiate into male and female gametocytes, which are the sexual stages of Plasmodium. These gametocytes can be taken up by another female Anopheles mosquito when it feeds on an infected human. Inside the mosquito's gut, the male gametocyte produces 8 flagellated microgametes that fertilize the female gametocyte, forming a zygote. The zygote then develops into an ookinete, which penetrates the gut wall and forms an oocyst under the gut epithelium. The oocyst grows and produces thousands of sporozoites by a process called sporogony. The oocyst then ruptures and releases the sporozoites into the mosquito's hemolymph, body fluid, where they migrate to the salivary glands and wait for the next opportunity to infect a human host. When the mosquito bites another person, it injects some saliva along with the sporozoites into the bloodstream. The sporozoites then make their way to the liver, where they multiply and start a new cycle of infection. This is how malaria is transmitted by mosquitoes, but did you know that there are other ways that people can get malaria? Although rare, malaria can also be passed from mother to child during pregnancy or delivery. This is called congenital malaria, and it can cause serious complications for both the mother and the baby. Malaria can also be transmitted through blood transfusion, organ transplantation, or sharing of needles and syringes. However, these modes of transmission are very uncommon in countries where malaria is not endemic. So now you know how malaria spreads and why it is important to prevent mosquito bites and use safe blood products. Next in this video, I will talk about pathogenesis, how malaria can be diagnosed, and different types of malaria. Pathogenesis. Picture this: a warm summer evening, a gentle breeze, and unfortunately, a hungry mosquito lurking nearby. If that mosquito happens to be carrying the malaria parasite, what happens next? Well, that's where the story of pathogenesis begins. When a mosquito infected with the malaria parasite bites a person, the individual can experience different outcomes. There are 4 possible scenarios. First, the person might not get infected at all. Second, they may have an asymptomatic infection, where the parasites reside in their bloodstream without causing any noticeable symptoms. Third, they could develop uncomplicated malaria, characterized by symptoms and the presence of parasites, but without major organ dysfunction. Lastly, the person may suffer from severe malaria, where symptoms, parasitic infection, and significant organ dysfunction occur. Now, you might be wondering, what determines which outcome a person will experience? Well, there are several factors at play. Let's start with the parasite factors. The species of the malaria parasite, the number of parasites injected, their virulence, and drug resistance can all influence the clinical outcome. On the other side, we have the host factors. Age, pregnancy status, immune status, and genetic polymorphisms all play a role in determining the severity of the disease. Lastly, we have geographic and social factors. Endemicity, poverty, and access to treatment in a given area can also contribute to the clinical outcome. In regions with high Plasmodium transmission, most people develop immunity over time. However, severe malaria remains most common among children under 5 years old and pregnant women. On the other hand, in areas with low transmission or among travelers to endemic regions, people of all age groups are at risk for severe malaria. It's important to note that severe malaria is predominantly caused by Plasmodium falciparum, but other species such as Plasmodium vivax and Plasmodium knowlesi can also lead to severe disease. In contrast, infections with Plasmodium malariae or Plasmodium ovale typically result in uncomplicated and benign malaria. Severe malaria is usually diagnosed when more than 2% to 5% of red blood cells are parasitized. But what are the complications associated with this severe form of the disease? Well, let me tell you, severe malaria doesn't hold back. It can lead to respiratory distress syndrome, metabolic acidosis, circulatory shock, renal failure, hepatic failure, hypoglycemia, severe anemia, poor pregnancy outcomes, and even cerebral malaria, a potentially life-threatening condition affecting the brain. Anemia often accompanies malaria. However, the causes of this association are far from simple. Let's uncover the multifaceted factors that contribute to this intriguing connection. As the malaria parasite progresses through its growth cycle, it directly targets and destroys infected red blood cells. But here's where it gets interesting: the parasite also triggers immune responses that lead to the destruction of both infected and non-infected red blood cells, primarily within the spleen. This immune-mediated destruction of non-infected cells significantly contributes to the development of anemia. But the story doesn't end there. During the invasion, the malaria parasite sheds proteins that alter the membranes of non-infected red blood cells, making them more susceptible to spleen-mediated removal. It's almost as if the parasite has devised a cunning strategy to exacerbate the impact on red blood cell populations. Furthermore, parasite proteins can induce oxidative damage, leading to reduced red blood cell survival. This oxidative stress further contributes to the decline in red blood cell count and exacerbates anemia. Malaria parasites also during the metabolization of hemoglobin, resulting in the formation of toxic byproducts called hemozoin or malaria pigment. When red blood cells rupture during schizogony, these toxic metabolites, along with other substances, are released, triggering an inflammatory response and causing an imbalance in cytokine levels. The abnormal levels of cytokines, such as tumor necrosis factor alpha and interferon gamma, can inhibit erythropoiesis, leading to ineffective erythropoiesis. This disruption in red blood cell production further contributes to anemia in individuals affected by malaria. But here is the twist. Increased levels of interleukin-6, stimulated by the malaria infection, stimulate the production of hepcidin in the liver. Hepcidin, in turn, reduces the availability of iron, a crucial component for red blood cell development. This scarcity of iron compounds the challenges of anemia in malaria-infected individuals. Moreover, factors such as poor nutrition and co-infections with parasites like hookworm or diseases like HIV, inflammation, and cytokine imbalances can significantly contribute to anemia in regions where malaria is endemic. These additional factors intricately intertwine, amplifying the complexities of battling malaria-induced anemia. Plasmodium falciparum, a particularly dangerous strain of malaria, exhibits a chilling ability to make infected red blood cells adhere to the walls of small blood vessels within various internal organs. This perilous phenomenon occurs in the brain, heart, lungs, liver, kidneys, dermis, and placenta. The consequences are dire. These blockages impede the normal flow of blood, reducing the vital oxygen supply to the affected organs. Additionally, this adhesion mechanism offers protection to the parasite, as it becomes less susceptible to clearance by the spleen's immune response. In pregnant women, this gripping of infected cells in the placenta can lead to severe anemia, hinder fetal growth, and elevate the risk of fetal loss. In the brain, the repercussions are even more sinister, culminating in a life-threatening condition known as cerebral malaria. The infected red blood cells bear a protein on their surface called PFEMP1, enabling them to bind to specific receptors on blood vessel walls, including CD36 found on platelets and certain endothelial cells. While scientists continue to unravel the mysteries behind the severity of cerebral malaria, it has been discovered that a variant of PFEMP1 can bind to a receptor called endothelial protein C receptor EPCR present in the brain's microvessels. This binding process inhibits the activation of a vital protein called protein C, which typically regulates the activation of factors 5 and 8. Consequently, a hypercoagulable state ensues within the brain, leading to the deposition of fibrin and the sequestration of parasites in the microvasculature, triggering the distressing symptoms of severe cerebral malaria. The ability of the malaria parasite to invade red blood cells plays a pivotal role in determining the severity of the disease. While Plasmodium vivax and Plasmodium ovale exclusively invade immature red blood cells, reticulocytes, Plasmodium malariae solely targets older red blood cells. In stark contrast, P. falciparum and Plasmodium knowlesi exhibit the ability to invade red blood cells of all ages, leading to significantly higher parasitemia and more severe disease outcomes. It is worth noting that Plasmodium vivax's invasion depends on the presence of Duffy antigens on red blood cells. Consequently, individuals without Duffy antigens exhibit resistance to Plasmodium vivax infection, a genetic adaptation that has proven effective in regions like West Africa, where Plasmodium vivax is nearly non-existent. Strikingly, certain genetic variations such as sickle cell trait, thalassemia, and glucose 6-phosphate dehydrogenase, G6PD, deficiency can confer protection against severe falciparum malaria, even in individuals who can still become infected. Clinical and laboratory findings play a crucial role in diagnosing and assessing the severity of malaria. Fever, chills and rigors, sweating, headache, muscle pain, nausea, and diarrhea. In severe cases of malaria, additional clinical findings may manifest. Jaundice, splenomegaly, hepatomegaly, shock and prostration, bleeding, seizures or coma. Laboratory investigations provide valuable insights into the severity and complications of malaria. Metabolic acidosis, increased acidity in the blood due to impaired metabolic processes. Decreased serum glucose by less than 40 mg per deciliter. Increased serum lactate, indicating tissue hypoxia. Increased serum creatinine. Decreased hemoglobin level of less than 5 grams per deciliter indicating anemia, hemoglobinuria, hyperparasitemia. In patients with chronic malaria or repeated malarial infections. Splenomegaly. Decreased reticulocytes count, attributed to the negative impact of chronic inflammation on erythropoiesis. White blood cell count during fever episodes, the WBC count is typically normal to slightly increased. However, during chills and rigors, neutropenia may develop. When it comes to diagnosing malaria, the microscope becomes our trusty companion. Before treatment begins, it's crucial to collect blood samples from individuals suspected of having malaria. These samples, obtained from veins, hold the key to unraveling the presence of those pesky parasites. To ensure accurate results, the collected venous blood is mixed with a special anticoagulant called ethylene-diamine tetrachic acid or EDTA. This prevents the blood from clotting, allowing us to work with it more effectively. Now, let's get creative. The mixed blood is used to create 2 thick and 2 thin peripheral blood films. Think of these films as our canvas, waiting to be painted with insights about the presence of malaria parasites. To bring the parasites into clear view, we use a remarkable stain called Reichgemse. This water-based stain is like a magician's wand, revealing the parasites in all their glory. The thick blood films are particularly useful for initial screening, while the thin blood films help us identify the species and measure parasitemia. Now comes the detective work. Using a thin blood film, the experienced lab personnel meticulously count the number of parasitized red blood cells among hundreds of regular red blood cells. This count gives us an idea of the percentage of parasitemia, which tells us the severity of the infection. With their skilled eyes, the lab experts examine at least 300 fields on both the thick and thin blood films using a powerful 100x objective lens. This thorough examination ensures we don't miss any hidden parasites lurking in the blood. Here's an important tidbit: the number of circulating parasites can vary depending on the timing of erythrocytic schizogony, the parasite's life cycle stage. That's why multiple samples may be necessary, taken at intervals of 8 to 12 hours, to capture the full picture. Before we wrap up, remember that detecting malarial parasites and species identification require experienced laboratory personnel. Inexperienced observers can sometimes mistake a platelet lying on top of an erythrocyte in a thin blood film as a malaria parasite. Plasmodium vivax is responsible for about 40% of malaria cases globally. It primarily haunts regions across Asia, South and Central America, Southeast Asia, Oceania, and the Middle East. Surprisingly, it is rare in Africa and notably absent in West Africa. When it comes to identifying this elusive parasite, the microscope becomes our ultimate tool. Plasmodium vivax reveals its presence through delicate early ring forms. Look closely, and you'll notice a distinctive red chromatin dot within the blue staining cytoplasm. These unique characteristics give us a clue to its identity. As the parasite grows within the red blood cells it infects, something fascinating occurs. The infected cell becomes enlarged, and a phenomenon known as Schaffner stippling appears. This stippling can be observed in all stages except the early ring forms. As the life cycle progresses, the Plasmodium vivax trophozoite matures and nearly fills the red blood cell it occupies. And when it's time for reproduction, the schizont bursts, releasing an astonishing 12 to 24 merozoites into the bloodstream. It's a true spectacle of nature. But Plasmodium vivax doesn't stop there. It also produces round gametocytes with a distinguishing blue cytoplasm and light brown pigment. These gametocytes play a crucial role in the transmission of the parasite from one host to another. Now, here's where things get even more intriguing. Plasmodium vivax is notoriously difficult to eradicate because it has a hidden ace up its sleeve. It forms hypnozoite forms, lying dormant in the liver. These dormant forms can awaken and cause relapses, making complete elimination a formidable challenge. Plasmodium malariae, while relatively rare, can be found in various parts of the world. Its highest prevalence is observed in East Africa and India, where it has left its mark on the local populations. By carefully observing blood samples under the microscope, we can unravel the distinct features that set it apart from its counterparts. The early ring stage of Plasmodium malariae is a fascinating sight to behold. These early forms are usually smaller and wider compared to those of other malaria parasites, such as Plasmodium vivax. However, it can be quite challenging to differentiate between the two at this stage. As the Plasmodium malariae trophozoite grows within the red blood cell, a unique characteristic begins to emerge. The cytoplasm forms a narrow band across the cell and contains dark brown pigment. This distinct feature aids us in identifying this particular species. Unlike its counterparts, Plasmodium malariae does not cause enlargement of the red blood cells it infects, nor does it exhibit the characteristic stippling we observe in other species. These differences provide further clues in the identification process. When it comes time for reproduction, Plasmodium malariae's mature schizoont releases 6 to 12 merozoites into the bloodstream. These merozoites, though similar to those of Plasmodium vivax, are smaller in size. It's incredible how even within the microscopic world, diversity exists. And let's not forget about the gametocytes of Plasmodium malariae. These fascinating forms often gather and form a rosette around clumped pigment, adding another layer of uniqueness to their appearance. Plasmodium falciparum is predominantly found in sub-Saharan Africa, where it poses a significant health challenge. However, its reach extends beyond Africa, touching regions such as Saudi Arabia, Haiti, the Dominican Republic, parts of Asia, Southeast Asia, the Philippines, Indonesia, and South America. When examining blood samples under the microscope, Plasmodium falciparum presents itself as ring forms and crescent-shaped gametocytes. The ring forms may be challenging to detect as they are small and can even appear as multiple rings within a single red blood cell. Their cytoplasm takes on a deep blue hue, accompanied by brownish pigment and red chromatin near the center. Truly a captivating sight. What sets Plasmodium falciparum apart is its remarkable ability to adhere to endothelial cells in various organs instead of freely circulating in the bloodstream. This unique behavior contributes to the severity of the disease and adds another layer of complexity to its life cycle. Time is of the essence when it comes to Plasmodium falciparum's life cycle. The erythrocytic cycle of this parasite lasts between 36 to 48 hours, showcasing its efficiency in invading and multiplying within red blood cells. Plasmodium knowlesi, a species of the malaria parasite, calls the vibrant landscapes of Malaysia its home. It has also made its presence known in neighboring countries such as Myanmar, the Philippines, and Thailand. In its early trophozoite stage, Plasmodium knowlesi shares similarities with the notorious Plasmodium falciparum. It displays high parasitemia, with multiple ring forms within a single red blood cell. These early stages can be quite deceptive. As the trophozoite stage progresses, Plasmodium knowlesi sets itself apart. Its cytoplasm forms a distinct band across the cell, reminiscent of another malaria parasite, Plasmodium malariae. Mature schizounts of Plasmodium knowlesi bear an average of 16 merozoites and do not form rosettes, unlike other species. These distinctive traits aid in distinguishing this captivating parasite. One key factor that sets Plasmodium knowlesi apart is its short erythrocytic cycle of only 24 hours. This rapid multiplication within red blood cells can lead to high parasitemia and severe malaria in infected individuals. While microscopic examination provides valuable insights, definitive diagnosis of Plasmodium knowlesi may require molecular methods. These advanced techniques play a crucial role in confirming the presence of this remarkable parasite. Now it's your turn. Comment below, which species do you find most challenging? Share your thoughts and experiences. Don't forget to subscribe, activate notifications, and share this video with your friends. While microscopic examination is a valuable tool, there are alternative methods that provide additional insights. Let's explore some of these diagnostic approaches. Fluorescent dyes offer a means of detecting the presence of Plasmodium parasites. Although they don't identify the specific species, they provide a reliable indication of infection. However, when it comes to species identification, we need to turn to molecular tests. Molecular tests, such as polymerase chain reaction, PCR, revolutionize malaria diagnosis. These tests not only detect the presence of the parasite but also identify the specific species. This is especially crucial in cases of mixed infections, low parasitemia, and infections with elusive species like Plasmodium knowlesi. For rapid and convenient diagnosis, we have the BinaxNOW malaria test approved by the FDA. This antigen test detects the presence of Plasmodium falciparum histidine-rich protein 2 and generic Plasmodium aldolase. It offers quick results, aiding in timely intervention. However, it's important to note that rapid antigen tests like BinaxNOW have their limitations. They may have lower sensitivity when the parasite count is low, typically below 100 parasites per microliter of blood. To overcome this, it's recommended to combine antigen tests with traditional microscopic examination using thick and thin blood films. Babesiosis, a unique disease transmitted by tiny yet mighty creatures known as ticks. Join us as we unravel the secrets of this fascinating illness and explore its intriguing aspects. Babesiosis is caused by a group of parasites called Babesia, which are transmitted through the bite of infected ticks. While there are many species of Babesia, only a select few can cause disease in humans. In the United States, the most common cause of babesiosis is Babesia microti. This particular species was first identified on Nantucket Island in Massachusetts during the late 1960s and early 1970s. The disease cycle of Babesia microti occurs within two primary hosts, the tick, Ixodes scapularis, and the white-footed mouse, which acts as a reservoir. The sexual cycle of the parasite takes place within the tick, while the asexual cycle primarily occurs in the white-footed mouse. Humans become accidental hosts of Babesia when they are bitten by infected ticks. It's important to note that although rare, other Babesia species, such as Babesia duncani and Babesia divergens, can also cause disease in humans. Interestingly, Babesia can even be transmitted through blood transfusions. There have been cases of transfusion-transmitted babesiosis, highlighting the importance of screening blood donations. Furthermore, while rare, babesiosis can also be passed from a pregnant woman to her child, underscoring the need for caution and awareness. Now, let's delve into the clinical findings associated with Babesia microti infection. While the incubation period can vary from 1 to 9 weeks, the range of symptoms and their severity is quite diverse. Some individuals may remain asymptomatic, while others may experience a wide array of flu-like symptoms. These can include fever, chills, headache, sweats, nausea, joint pain, muscle aches, loss of appetite, and fatigue. These symptoms may persist for a few weeks to several months. In some cases, individuals may exhibit additional clinical findings. These can include jaundice, which presents as yellowing of the skin and eyes, as well as splenomegaly, an enlarged spleen, or hepatomegaly, an enlarged liver. While most individuals recover without complications, it's important to note that some cases can progress to severe, life-threatening diseases. This can involve various organ systems, such as acute respiratory failure, congestive heart failure, renal shutdown, liver failure, central nervous system involvement, or disseminated intravascular coagulation. Severe disease can affect individuals of any age, but it tends to be more common in those over 50 years old. Additionally, Babesia microti infection can lead to mild to severe hemolytic anemia. When it comes to diagnosing babesiosis, laboratory findings play a crucial role in providing valuable evidence and insights into the infection. Let's explore some of the key laboratory findings and diagnostic methods used by doctors. One of the significant indicators is hemolytic anemia, which can be observed through laboratory tests. These may reveal a decrease in hemoglobin levels, an increase in reticulocytes count, a decrease in serum haptoglobin levels, and the presence of bilirubinemia. Additionally, patients with babesiosis may exhibit other abnormalities in their laboratory results. These can include leukopenia, thrombocytopenia, hemoglobinuria, proteinuria, as well as abnormal results on renal and liver function tests. To make a definitive diagnosis, doctors rely on microscopic examination of Röntgen-sustained thin peripheral blood films. Babesia parasites, named after the disease, appear as small rings or occasionally as tetrads within red blood cells. These parasites can take different forms, such as round, oval, or amoeboid, with a dark purple chromatin dot and minimal blue cytoplasm surrounding a vacuole. Multiple rings can be seen within a single red blood cell, and tetrads may form a distinctive Maltese cross formation. These features help distinguish Babesia from other parasites, such as Plasmodium falciparum. In cases of low parasitemia, diagnosing babesiosis can be challenging through direct observation alone. In such instances, the detection of specific antibodies can be achieved using an indirect immunofluorescent antibody assay. This test identifies the presence of IgG and IgM antibodies to Babesia microti, with a sensitivity ranging from 88% to 96%. For definitive species identification and to differentiate between different Babesia species, PCR-based methods are employed. These molecular techniques analyze the genetic material of the parasite, providing accurate and specific results. Clostridial sepsis, a grave condition that can have devastating consequences, is caused by the anaerobic gram-positive bacillus known as Clostridium perfringens. Let's dive deeper into this condition and its associated features. Clostridial sepsis occurs when the rapidly growing Clostridium perfringens releases a toxin called alpha toxin. This potent toxin targets the phospholipids in the red blood cell membranes, leading to a catastrophic event, fatal intravascular hemolysis. The sudden and massive hemolysis caused by the alpha toxin results in distinctive clinical manifestations. The plasma and urine take on a dark red color, and the hematocrit, a measure of red blood cell volume, drops precipitously below 10%. This severe hemolysis can also trigger a cascade of complications, including disseminated intravascular coagulation, DIC, and renal failure. When examining peripheral blood films under the microscope, certain characteristic changes may be observed. Spherocytes, microspherocytes, and toxic changes to neutrophils can indicate the presence of Clostridial sepsis. Clostridial sepsis and hemolytic anemia are more likely to develop in individuals with specific predisposing factors. These may include malignancies, solid organ transplantation, postpartum or post-abortion infections, biliary surgery, acute cholecystitis, and deep wounds. Prompt and aggressive treatment is crucial in managing Clostridial sepsis, as the prognosis is generally poor. Rapid interventions, such as transfusions to replenish blood loss, administration of appropriate antibiotics, and careful fluid management, are essential in the battle against this life-threatening condition. Despite intensive treatment efforts, many patients sadly succumb to the severity of Clostridial sepsis, highlighting the urgency and importance of early detection and intervention. Bartonellosis, a fascinating and unique disease also known as carrion disease. Let's embark on a journey to uncover the mysteries surrounding this condition. Bartonellosis is predominantly found in specific regions of Peru, Ecuador, and Colombia, where it is transmitted through the bite of female sandflies. The disease is caused by a bacterium called Bartonella bacilliformis, which has a peculiar affinity for red blood cells. It attaches to these cells and triggers a process known as hemolysis, where the red blood cells are destroyed. One of the intriguing mechanisms employed by this bacterium is the production of a protein called deformin. Deformin creates pits or invaginations in the membrane of the red blood cells, contributing to their destruction. Bartonellosis manifests in two distinct stages. The first stage is the acute phase, characterized by acute hemolytic anemia, while the second stage, known as the chronic phase, is marked by the appearance of skin lesions and warts on the body. During the acute phase, individuals may experience symptoms such as fever, malaise, headache, and chills. As the disease progresses, pallor, jaundice, and lymphadenopathy may become apparent. Rapid hemolysis takes place during the acute phase, leading to a significant decrease in hematocrit levels. Sadly, the mortality rate for untreated cases is high. To diagnose Bartonellosis, blood cultures are conducted, and a Röntgen-sustained peripheral blood film is examined. This enables the observation of bacilli or coccobacilli on the surface of the erythrocytes. Treatment for Bartonellosis involves a multifaceted approach. Transfusions are administered to replenish the lost blood, while antibiotics such as ciprofloxacin or chloramphenicol are prescribed to target the bacterium. It is intriguing to note that the disease was named after Daniel Alcides Carrion, a Peruvian medical student who, in 1885, deliberately infected himself with fluid from a wart of a patient in the chronic stage of the disease. His courageous act contributed significantly to our understanding of this condition. Hemolytic anemia caused by red blood cell injury resulting from drugs and chemicals. Let's uncover the mechanisms and factors involved in this fascinating phenomenon. Certain drugs and chemicals have the potential to damage red blood cells, leading to hemolytic anemia. They achieve this by triggering oxidative denaturation of hemoglobin, a process that generates substances such as methemoglobin and Heinz bodies. The severity of hemolytic anemia caused by these agents can vary. Some drugs, like dapsone, used in the treatment of leprosy and dermatitis herpetiformis, as well as naphthalene, a chemical found in mothballs, can induce hemolytic anemia in individuals with normal red blood cells. However, individuals deficient in glucose 6-phosphate dehydrogenase, G6PD, an enzyme crucial for protecting red blood cells from oxidative stress, are particularly susceptible to the effects of these oxidative agents. For example, in individuals with G6PD deficiency, the drug primaquine can cause hemolytic anemia. Primaquine is commonly used in the treatment of malaria. When assessing drug or chemical-induced hemolytic anemia, specific laboratory findings can provide valuable insights. These include a decrease in hemoglobin levels, an increase in the reticulocytes count, an elevation in serum indirect bilirubin, and a reduction in serum haptoglobin levels. In more severe cases, Heinz bodies may be observed within red blood cells. These bodies, denatured hemoglobin aggregates, can only be detected using a supravital stain. They appear as round, blue granules attached to the inner membrane of the red blood cell. Additionally, exposure to high levels of substances such as arsenic hydride, copper, and lead can also instigate hemolysis, further contributing to the development of hemolytic anemia. Understanding the underlying mechanisms and laboratory findings in drug or chemical-induced hemolytic anemia is essential for accurate diagnosis and appropriate management of affected individuals. By identifying and mitigating the factors that contribute to red blood cell injury, we aim to improve patient outcomes and prevent further complications associated with this intriguing form of anemia. Hemolytic anemia resulting from venomous bites or stings can occur immediately or manifest after a delay of 1 or more days. The severity of the hemolysis is determined by the amount of venom injected, and in severe cases, it can lead to renal failure and even prove fatal. Venoms have various mechanisms through which they can induce hemolysis. Some venoms directly damage the red blood cell membrane, causing its disruption and subsequent release of hemoglobin into the bloodstream. Other venoms alter the red blood cell membrane in a way that triggers complement-mediated lysis, a process involving the immune system's complement system that leads to destruction of red blood cells. Furthermore, certain venoms can activate disseminated intravascular coagulation, DIC, which can contribute to hemolysis and other complications. Hemolytic anemia resulting from venomous bites or stings has been reported in various scenarios. Some venomous snakes, such as certain cobras and pit vipers, have been known to cause hemolysis in affected individuals. Additionally, bites from spiders like the brown recluse spider, Loxosceles reclusa and Loxosceles lata, have been associated with hemolytic anemia. Multiple stings, typically 50 or more, by bees or wasps can also induce hemolysis in susceptible individuals. It is crucial to recognize the potential for hemolytic anemia in venomous bites or stings and seek prompt medical attention to mitigate the risks and manage potential complications. By understanding the mechanisms and consequences of hemolysis caused by venoms, we can enhance our ability to provide appropriate care and safeguard the well-being of those affected. In our exploration of hemolytic anemia, we now delve into an intriguing connection between extensive burns and the disruption of red blood cells. Thermal injuries, particularly extensive burns, can lead to acute hemolytic anemia due to the injury inflicted upon red blood cells. When normal red blood cells are exposed to temperatures of around 49 degrees Celsius, or 120 degrees Fahrenheit, in laboratory conditions, they undergo a process known as fragmentation. This causes the cells to break apart into fragments and buds. Similarly, patients with extensive burns may experience acute hemolytic anemia as a result of red blood cell injury. The peripheral blood film of these patients often reveals the presence of schistocytes, spherocytes, and microspherocytes, indicating the fragmentation and altered shape of red blood cells. Although the damaged red blood cells are typically cleared by the spleen within 24 hours of the burn injury, the anemia associated with extensive burns can arise from various factors. Firstly, blood loss during surgical procedures performed to treat burn injuries can contribute to anemia. Additionally, nutritional deficiencies, impaired metabolism, and anemia of chronic inflammation further exacerbate the condition. Furthermore, in unfortunate cases, malfunctioning blood warmers used to heat blood prior to transfusion can cause overheating, leading to the fragmentation and hemolysis of donor red blood cells. The treatment of hemolytic anemia resulting from extensive burns requires a comprehensive approach. It involves addressing the underlying causes, managing blood loss, correcting nutritional deficiencies, supporting metabolic function, and addressing any chronic inflammatory processes. By understanding the impact of extensive burns on red blood cells and recognizing the contributing factors to anemia, healthcare providers can develop tailored treatment plans to support the recovery of patients. Stay up to date with our latest videos by hitting that subscribe button and activating notifications. Be the first to know when we release new and exciting content. Don't keep all this valuable information to yourself, share this video with your friends who might find it interesting and beneficial. If you have any questions or comments, feel free to leave them down below. Until next time, take care and goodbye.